So just to set the stage, everyone here is intimately familiar with a lot of these trends, but always good to contextualize stuff. Illumina claims to somehow generate 5 billion single cells over the next 3 years. We'll see if that happens. We have, the wonderful Tahoe, the Arc Institute and the BioHub planning to generate 130 million cells over the next year or so. And in industrial research these days, we regularly see, over 1 million cell atlases for routine screening or surveying of tissue types. So something's happening. I come to you with the claim that it might actually be important to understand how assays work when doing machine learning. So the final integer value in your count matrix comes from a long chain of steps, both in the wet lab and on the computer. You have a molecule in the cell, you do reverse transcription, you do PCR amplification, the sequencer outputs reads, you group the reads by barcodes, and so on and so forth. You end up with this object that is becoming the more or less the primitive and modern AI ML, but the steps and information behind how that object was constructed is becoming increasingly important when you make modeling assumptions. So as we enter into this world of models and agents, they need to understand molecular capture. Here I have Claude Agent SDK, which is going to be one of the vehicles by which I think people parse data. And then we also have, again, Tahoe's perturbational model from a few years back, representing molecular foundation models, another avenue by which people might query this data. This question of what does a zero mean is still salient to them as it was to us, and perhaps more so. It can mean all these different things. Incomplete capture, reverse transcription efficiency, and so forth. So hence this themed meeting for single cell data, so we can start to poke and prod with a focus on the assay type. We're going to do a few more of these. So this is the first in a family of these kind of meetups. Quickly about us. So we build the agent infrastructure for large assays. Let me get this out of the way. Sorry. With the basic understanding that as data volume goes up, biology research will need both data infrastructure, so systems and cloud software to allow the storage and transformation of file data from increasingly large experiments, and agents, which are becoming, we think, soon the default way that scientists will interact with data. These are basically language models and loops that steer analysis of natural language. There's a strong interaction between 2 and 1. And 1 is a unique component of biology that is not present in domains like code that many of you are working on, which is tool calls and operations actually can take days to weeks, and terabyte of RAM, and a lot of pretty gnarly cloud expertise to get to run. And then the last thing I just want to point out is we really believe that measurement-driven agent engineering is the path forward. So there's a lot of vibing going on, but what you can measure, you can improve. And rigorously breaking down what scientific performance means in agentic context is one of our goals. And we've published work in single cell and spatial, and made this available both on our archive and on our benchmarks.bio resource. But we're expanding rapidly to new domains, and are hiring technical staff, so engineers and scientists across new categories in epigenetics, genetics, proteomics, of course, single cell, diagnostics, translational assays, including PK, PD, and immunogenicity, and so forth. Okay, so that's enough about the backdrop. Now I would love to introduce our wonderful speakers. Valentin from Tahoe is going to be talking about really cool, deep work on effective drug screening from his time parsing and modeling enormous volume of single cell counts. Michaela from Assayverse is going to be talking about a variety of techniques to make data infrastructure agentic in the single cell context and perhaps spatial context. And then Hari and Jen, both technical staff at Latch, have been hard at work deep in the guts of single cell data, trying to figure out how you measure frontier model performance when is trying to analyze this stuff. So without further ado, Valentin, here you go. Thank you.
Thank you so much for inviting me. This is awesome, and this is a good opportunity to gather together, I guess, here and talk more technical a bit. I think I'm going to be more high level, but I'm more than happy to take questions lower level. I will introduce Assay for those who don't know, and talk briefly about what we're doing and how we're trying to prepare this era of AI. Because I figured that a lot of people use tools that they belong in our organization, they're maintained by us, but they're not necessarily aware who we are and what we're doing. It was a very good introduction before, because we mentioned the AI boom that just passed, and now that the data boom that was announced by the latest JPM. And my thinking is, OK, people are generating massive amounts of data. What is coming next? It should be the infrastructure, the data loading boom. How do you load this data? We have all these GPUs, CPUs nowadays, all this infrastructure happening, and a huge amount of data, but these necessarily are not talking to each other, not properly. So yeah, Assay is in this space. So I'm going to go a bit back and also briefly talk about 15 years of history, more or less. I'm Michaela. I'm a postdoc at Genome Tech in the morning. That's my actual job at Marioni, John Marioni's lab. And I'm a steering council and core member at Assay, which is the evening job the last 4 years, after my morning one. Doesn't pay my rent, otherwise, in other words. Story time, why do we exist? So 13 years ago, for those who don't know, there was the methods of the year was awarded by Nature Methods, and it was the single-cell RNA sequencing, which was not when it was actually happened. It was when it was established enough to be awarded as the methods of the year. And very soon after that, like 7 years later, 2020, we have the spatial transcriptomics as the method of the year by Nature Methods. And basically what was in the space, I'm trying to kind of move briefly from that because it's history, that's very interesting. So in the space was bioconductor, right? There was already most of the omics data history was in R. Statistics were there, and actually our story comes from there. We copy a lot of information from them. It was pioneers. So they have all these summarized experiment, multi-assay experiment, single-cell experiment. Because the space was kind of fragmented, this starts showing the way of how do we gather the community, how do we gather the tools, and how do we take advantage of this data. So in a similar effort, there was Undata happened 2018, and actually ScamPy even before that, which was 2017, published in 2018, but was existed. It was an effort from Alex Wolf and Thijs lab in Munich because they didn't like R back then. No hard feelings. I like both. But it was an effort. And they were trying to establish there the ScamPy, which was a similar effort in R. But it was a very fragmented space. And again, you basically create another issue where you added an extra tool that communicates with nothing. You had to translate the data and transfer them and everything. So Undata, which is annotated data, came to save this and create an object where you load your data properly and you store all the information you need. And soon after that, 2020 officially we were launched, and it was the Assay, which is basically the ecosystem around that. And I will explain, this is a very preliminary, the first publication we had where we're establishing the ecosystem, and it's already outdated because already the data structures are more than this one. But it kind of captures what we do and who we are. In our heart is the data structures. So we have the Undata for single cells and MU data for multiomics, and nowadays spatial data too for spatial data. And the outside layer is the Assay core packages. You may recognize some of them, but basically the idea is that they are all the preliminary steps to analyze the data that they are coming from the data structures. So the first step is data loaders, data structures. Second step, core packages to analyze this data, the preliminary analysis. So the difference with the ecosystem is that these two layers we maintain and we develop ourselves. So the second part, the ecosystem, is all the tools that they actually comply with our code of conduct. They have some very specific minimal requirements to take advantage of our data structures, basically, and be in the space of single cells. But they are maintained by the developers. We only work with them just to make sure that it's easily maintained, the tools. And if some tools become very important for the community, we can actually take over and put them in the core, in collaboration always with them. And the outside part is always the community. We're very community-driven. This is how we exist, and we do all the events and everything. So data structures, I introduced them. This is, you're familiar with the space. Undata annotated is basically the main way to load your data in an object right now in Python. Undata, MU data is a dictionary of undatas, so as many modalities as you like. They just share metadata, and they are basically structured and connected together. And spatial data is kind of interesting because it has undata, the tables, the expression values are in an undata object, but you can also load all the pointers, like the images from raw images are and everything. You can all load it in spatial data, and they are layered and stacked, so you can analyze them together, and you can maintain all the information. And the core packages that we have is right now, because we expanded recently also, ScanPy for all the single cell preprocessing up to visualization, basically, even differential expressed genes. First steps you can do with ScanPy, SquidPy for spatial data, similar idea. The same expansion of preprocessing of multiomics data is the muon. SCVI tools, Valentine is one of the main contributors, not directly probably in SCVI, right? But all the ecosystem around that. So it's the variance inference tools for all things single cells. Snap attack for attack-seq, decoupler for inference, gene inference, ShearPy for immune repertoire, PertPy for perturbation data, and Rapids, which is very cool. Our rapid single cell is the wrapper of ScanPy, basically, but takes advantage of GPUs. We're talking about from hours to seconds. It's insane. It's one of our biggest tools at the moment, and we love it, and it's a collaboration with NVIDIA. But as I mentioned, a big part of Assayverse, for those who are interested in getting involved or getting to know us, is the community, because we're a community-led organization. And sometimes there's this misconception, because most of us, we're developers there, but basically developing and writing code is only one thing, and it doesn't come only from the developers, especially these days with agents. Writing the code is probably the least of our worries. But we do all these hackathons where community gathers together and solves all issues, PRs, pull requests in a way that we have open. And then we have all of the support on GitHub, tutorials, you name it, we do it. Atlases we maintain. And basically, we're inviting always the scientific community to contribute in all the ways. And you can do it from any position you have, either you're in company or academia, it doesn't matter. Any contribution helps in an open source organization. Some stats, just to flex a bit, because that's necessary. So, I mean, here we saw that we have more than, I don't believe that GitHub stars is a great way to evaluate tools. That's fine, though. We don't have better metrics. So on data, basically, you can see our biggest one is ScanPy, of course, makes sense, the GitHub stars, and also spatial data that are coming up, because it's a more recent addition. SCVI tools, of course, huge impact in the field. But the most important is, and this is kind of outdated now, we have more than a million monthly downloads of all of our tools, which is insane. And I will explain later why it's insane and what is the main issue we're facing. So this is increasing a lot. And nowadays with agents, please don't submit PRs that you haven't reviewed first. It's an issue, okay? Zulip, for those who don't know and would like to find us, Zulip is, I guess you know Slack. Zulip is Slack for cool kids, and we're the cool kids, so we do Zulip. Zulip is awesome. There are a lot of reasons we're there. A lot of open source communities are moving there right now. Bioconductor is there, NumFocus is there, Napari is there. So you have to join us. We have like way more right now than a K registered users and 1 million messages read, which basically means that we have like around 100, 150 people always interacting. So if you have any kind of question, there's going to be someone that will pick it up and will respond. And the developers are always there. So if you have a question about a tool, probably you will find them live to answer your questions. It's not a forum necessarily, but you can also use it like that. Right, flexing a bit more, just the things we've done with Assayverse, because the last 2 years we've been working very hard, I think, and we're producing very hard. We have collaborations with a lot of academic labs, and we are publishing papers in collaboration with them. Biocontext AI, that we'll briefly mention a bit later, is one of them. Undata R, so now the R users have no excuse anymore. They can use undata if they want, and they can transfer all their data back and forth in languages. We are in every space. We're doing it in Rust soon. Spatial proteomics, PertPy, and other spatial proteomics is 2 different papers. Ignore. And a second one. I mean, we have this consortium is very live. So we have a lot of efforts, and I like, before I go to the final topic, which is kind of more the AI and how do we make ready and the new efforts, I would like always to introduce the team somewhere in the middle where I kind of have attention or I get back the attention. The highlighted ones were the steering council. That doesn't mean anything, of course, for you. It's like we're part of the core. You can find a lot of us nowadays in, around here, San Francisco. And the rest, they are all over the world. This is an awesome team who has been together for years. And of course, our management committee, which helps us and funds most of the things so far. You may recognize some names, but basically we write grants together, they advise us during the process, and we meet very often with them. And finally, we have the advisory committee, which we meet a couple of times per year, and best case scenario. And if we meet, they are very, very helpful because they steer more of the directions that we have. But the most important is the contributors and the community, because I mean it for an open source, the most important is the community, and any kind of contribution really matters. Just for me to mention here already that from all the people you see, only 2 are full-time employees at Assayverse. The rest were volunteers. We don't get paid for that. It's very important for the rest. So where we are and what are we doing later. So I kind of skipped the first part. They are always very important. We have been doing them 2025, 2026, probably since we started 2022. We've been trying. We scaled more than 100 million cells to analyze. We do multimodal, any modality. Nowadays, we basically try to do all things single cell. We started as transcriptomics, but in reality, single cell is a resolution. We need all the modalities to understand the cells, to understand the drugs, how they interact. It's not only one version, especially the virtual cell that is a big thing nowadays. If you only consider RNA, it's a virtual mRNA now. It's not a virtual cell, in my opinion. So I think multi-omics is a big thing. We always care about benchmarking, and especially now we have a bigger effort coming up on benchmarking, and of course user accessibility. But lately we want to be the cool kids, as I said. So we always care about agents and Assayverse-specific MCPs. We've been collaborating with Anthropic, that they have been sponsoring and giving us some of the credits that they use they give for open source, and some power, mental power, I would say, not engineer power yet, but we've been discussing. And we have Into Context AI that I mentioned before, which is a company right now that they basically create all type of MCPs. It's kind of an effort between academia and nonprofit, but they are turning into profit. So don't quote me on that. You can cut it later, right? Right, in case it's too soon. And here is what I want to mention, what we're doing, because the idea is that we're not creating we are creating MCPs on data MCPs, or we collaborate on these things, but this is not what we're doing. This is not, we're infrastructure, right? We're trying to enable and make AI-ready data structures and data for people to be able to use them with LLMs. So the first thing that is kind of obvious, you would think, is that we need to make the data ready for AI, right? So the first thing we did is that we're working on this Unbatch, which is streaming data loaders in order to be able to saturate the models while training, from reading them from disk. Because as we said before, the models become bigger and bigger, so you cannot load them in memory, and you need to be able to train efficiently by loading from the disk. And that's kind of a solved problem. There was an ARC Institute effort recently in a similar vibe. So we did something very similar. Ours is undata tailored, so it should work directly with the whole ecosystem. But this is not the only thing that's important. This is happening, and it has to be a collaboration with companies as well, in my opinion, that they produce big data sets. The next thing that we care a lot is the metadata. So the data are important, and you can train models, and that's awesome, but it's always going to reflect the data, the annotation, right? How many things you know about the data and how well they are annotated. So we are building Croissant, awesome, metadata descriptors, which is a machine learning format, anyway, documentation for this job. So basically, now the tools can actually take advantage directly. They can store the metadata properly, and they can be read by the LLMs and agents, and not only, also in general machine learning models. And finally, we want to connect also everything with the existing cell ontology, the existing ontologies, especially MBL-ABI is our main target. We have cell ontology for cell types, Uberon for analysis, for anatomy, and disease ontology for conditions. In general, the main issue we solved with Unbatch is the input-output, because you end up a lot of times basically spending more time on loading your data than actually training them on the models. So that was the first goal. It's kind of solved. We're still working on it, but Unbatch is live. It exists. You can use it. And then another big effort, as I mentioned before, of course, is the actual MCPs and the agentic workflows. We already have an on-data MCP. You can use Claude. You can read it. It's live, or any MCP tailored anyway method. So you can use already and query your data. You can understand basically from on-data directly. And we are expanding this to spatial data, ScanPy, SquidPy, PertPy, basically all of our core packages. And this is indeed the collaboration that I was referring before with Anthropic, with the credits, and Biocontext AI, that they have released a lot of MCPs already. So now you can basically use directly on data, as I said, and continue with the rest when they come. But another thing that we care, I mean, not just us, and as I said, this is not our main goal, is how can you create a whole workflow, right? How can you give an on-data object now and basically get out of it, in our case, an Assayverse workflow, you would say, like use the Assayverse tools, but it doesn't matter. How can you get out the whole, do the batch correction, annotation, clustering, everything, and get the output to UMAP? I don't know. This is the very minimal workflow I can think of, or cell-cell communication. You can. So we're thinking of that. How can we tailor all the Assayverse tools and order them, or maybe pre-decide? It's kind of a tricky problem to solve, especially as a community, and it's not exactly what we're doing. But this is also in collaboration with Biocontext AI. Finally, another very, very exciting for the single-cell community thing that we've been doing is that we are rewriting, it's led by Rob Patry and James Ferguson. We're writing Star Aligner, which is, I don't know how many years. I don't know. I don't remember. It was written in C++ 98, the code base, and it's like more than 50K citations. It's the main tool for aligning a single-cell dataset, and it's purely unmaintained at the moment. So what we're doing is that it's called Rastar Aligner. It's also live. You can find it. And basically, we translate with Claude from Python, from C++ to Rust. And we already established that it kind of works. We're still running things. We need to make sure. But it's exciting. It's a collaboration with NVIDIA for the GPU accelerated components. But the cool thing is that it's a real-world example how we can use LLMs. I don't want to advertise only Claude, of course, but we are collaborating with them. And the main thing is, one of the problems that we face is the maintenance of tools, right? That's what we're trying to do, and that's what we're trying to provide for the community, but it takes a lot of effort. And this actually is the first demonstration that we may be able to do it fast, like quickly iterate, quickly maintain, and successfully, with minimal review. That's what we're trying to test here. So far it works. We'll let you know if things change. But this is exciting, actually, for us and for open source communities, I think, in general. So it's kind of a summary now, what's working. I briefly hyped our things. Unbatch works. On-data MCP is live. You can use it. Rastar Aligner also is happening. If there are any issues, you can also go on GitHub and contribute if you want. And Biocontext AI basically has created this skill to MCP breeds, where basically whatever MCPs exist, you can already take them as anything that reads skills. You can load them, the MCPs to the skills, and the skills can be loaded on the LLMs. It's also in collaboration with Claude, with Anthropic. But everything is not solved. And here I decided to put some open challenges. That doesn't mean I have the answer or I'm working towards that, just ideas. So, OK, of course, the first question is, how do we know biology is correct? I don't think it was solved as a humans. We solved that, and now the problem is the agents. I think this is a pure question. You run the whole workflow, and how do you know what you're running, the parameters you've chosen, what you see in the UMAP, or any other plot you generate is correct? The only issue now is kind of that, I don't know about you, but I don't really write code anymore in this analysis. There's no way I'm actually going to type anything in ScanPy anymore. They do it for me. And the problem is that you cannot review so much anymore. Like, the agents can decide to cluster, annotate, and then come up with some solutions, right? How do you even know? What is the benchmark? Not benchmark in a sense of biology necessarily, but how do I assess this in a way that the user will actually go and read the results? That's the main question. And then we have the issue of a lot of things now they're demonstrating some technologies, especially with agents and the pipelines. And then you have similar technologies. Here I demonstrate, I don't know, Visium, Murphy, Xenium, Cosmix, and Codex. that they don't, all the pipelines work from each other to each other, even though they should, they produce similar results. And that's, in my opinion, an issue because you want as general as possible, and then you need to think, why does it work? If the data are similar, why does it work in one and not in the other one, in the other modality? Metadata standardization at scale, as I said, garbage in, garbage out is not, I'm not the first one saying it, so it's very important, I think, to be able to curate the metadata. And as I mentioned, a big issue with all of these things is that a lot of things are relying on open source structures. Think about, we're talking about 1 million downloads per month, and a lot of companies, they are building on data and ScamPy. I bet among all of you, probably some of you use it, if not all of you. And as I said, well, there are only 2 employees, and the rest were volunteers. So this is not very sustainable, and that's a big issue and an open challenge. We're writing grants and everything, but yeah. And that's the final slide that I kind of repeat everything more or less, what we need next. I think I said it, I'm not gonna bother you more. I think is the data, they should be ready immediately to be read by the machines with metadata. They should be able to be loaded properly with GPUs, especially take advantage of GPUs and not just use them and like waste time on, the bottleneck is input output and then you have GPUs just waiting, then it's just a waste. So all of these things should be produced directly and documented for machines. And then basically we need verification, like we need reproducible agents. Right now all of us use them, but very individually, and you can't even share them and say, okay, to your collaborator, here it is, reproduce it entirely, or you're not sure basically. And finally, the sustainable. I would love the companies that they use us to find ways, we can find together the ways to sustain and sponsor us and help us grow and sustain ourselves. And I have a last message, because this event is single cells and our history is single cell RNA-seq. But this is food for thought. We've been expanding, as I said before, we're doing all things single cells, not just single cell resolution. Modalities are important. And lately, my obsession, you probably noticed from the papers that we published the last year, is proteins, because 70% of the drugs, they're actually predicted and validated on proteins, not on RNA. RNA is great, but just maybe not enough. And think beyond. That's food for thought, just on. Call for action, just contribute to the community. I mentioned everything, you don't need more than that. Anything you do is, even joining a hackathon is very important. And we have meetings also every other Tuesday. You should have introduced that, Valentine. And that's it, and thank you so much for inviting me, and I'm open for questions. How they look like? So the croissant metadata. So it's a very standardized, like a YAML almost file. It's standardized, but it's standardized in the sense that it's easy to be read by ML, machine learning models directly. And it's already pre-decided that it's tailored to the, if I can use English words for that. So it has predefined sections, and it's not very hard to use it. The hard thing with the metadata is to decide what you ask from the community to always have. We don't have that so standardized in single cell RNA-seq. Like, you should be able to ask, like, I don't know, Chromium, which version of which kit of 10x you used and everything to be always stored. And that's one of the efforts we're doing, to pre-decide what everyone needs to always claim in an undata object in order to be pre-processed. What do you need to store? And that has been a big issue, because now that we're moving to different modalities, each modality has their own requirements. So we're trying to find the bare minimum for everything.
How standard do you think you're going to be able to make metadata? Because I feel like it's so hard with all the different types of experiments people are doing to be able to do that.
Yeah. So the question is, how standard can we make the metadata, right? Yeah, we can't. So the idea of Assayverse in general is to always provide kind of best practices, right? We're infrastructure, and there's not one way to do things. I think that the impact we have right now in the community can be helpful to establish a way of doing it. And then if everyone does the same, I think that's at least how I see it, more or less. But if you have any suggestions, you are more than welcome to give us.
The idea of like rewriting libraries like Star with Rust sounds very promising. Like you said, it's really hard to maintain a lot of these tools. It's super interesting. What performance improvement did you see after rewriting it in Rust versus how it originally performed?
Yeah, that's a good question. I don't have the numbers entirely. The main thing it solves for us is not so much the speed, for example, or how accurate it is that we can maintain it now, right? Because the C++ 98 code base, it was not maintainable, and it was not maintained either. So probably you get some performance boost, for sure. I don't have the numbers, but I can look it up. But for us, it solves the maintenance problem, basically, as a community in general. Yeah.
Yeah.
Sorry, one last question.
Yeah.
How do you bridge the gap between metadata that's grounded in ontology versus, say, author-provided annotations or metadata? Because sometimes a newer paper might discover like a new cell state within a cell type, for example, but that might not necessarily exist in an ontology space. So what do you do in such edge cases?
Right, that's a very good question. One of the biggest discussions we have in the group is basically his question, which was, how do you solve? So annotations, you have already ontologies with some specific annotations, but most of the papers basically they discover new subtypes that they don't exist in the annotation, right? There's no entry. And the idea is, because we don't solve that, right? We offer solutions on how you can store this information properly. The idea is to connect the ontologies. So if it's actually justified, because I have also the feeling, I don't want to judge the science at all, but I have the feeling that a lot of times people claim things that they are, a set of markers that if you were looking in this ontology, maybe it was similar, whatever. So if there are some thresholds on some markers, it can be a new entry on your own data, and then it should be linked and published also in the ontology. So you populate an ontology, which is curated, because the EMBL EBI cell ontology is curated, right? So something like that we are thinking. But you should, as user, you should always have the flexibility to first use what exists, and then add anything you need on your data. And if you don't want to publish it and connect it with the ontology, that's up to you. But yeah, for us it's a dictionary, so you can have new entries. That's not an issue. Yeah. Cool. Thank you.
Thank you very much. We will probably donate. We seem to be active users of all the tools.
Active users?
Yeah.
He said we'll probably donate.
I didn't realize that you guys were taking contributions.
Yeah, for sure. Please contribute. Just not completely LLM generated PRs.
I meant more funding.
Funding. Yeah. Oh, you didn't know? Oh my God. So.
I didn't know there were only 2 full-time employees.
So yes, we are getting sponsorships, and we're changing now, for those who want to know, our non-profit. So we're going to have a board with companies, so we can talk.
Yeah, definitely. Last we have Hari and Jen. They're both really talented computational scientists here at Latch. They're going to talk about benchmarking single cell.
Good evening. I'm Hari, and along with Jen, who we're both part of the bioinformatics team at Latch. And today we're going to be presenting our work on Assay Bench, wherein we investigated frontier models' performance on a variety of single cell data analysis tasks. This was a joint work with Kenny and Hannah, who are right there rooting for us. So without ado, let me get started. All right, I'd like to start with this question, right? So can AI agents analyze real-world single-cell data? Yes. Sure. Well, reliably, let's see. Right, but that's the thought I want to leave you with, right? So basically, we have a ton of data that we've sequenced, and now analytical bottlenecks is kind of slowly beginning to creep up because we're not able to confidently say if the answer is right or not. And to that end, the AI agents have, to a large extent, been able to alleviate this problem, but I don't think we're there yet, and that's what we're trying to investigate with this experiment. So sequencing has definitely improved. The cost of sequencing has certainly gone down, but reliable analysis compresses the data through many fragile choices into a biological claim. And essentially, training these models to be able to confidently say where they can go wrong or like just even start by benchmarking their limitations is something that would definitely help us further how we think about these models. So to that extent, we looked at a variety of single-cell data sets. We had data, single-cell DNA data from Mission Bio's Tapestry kit that also has a protein panel. And we had Chromium data, Illumina's PIP-seq data, which is a single nuclei RNA-seq data, and data from BD Rhapsody, Pars Biosciences, Evercode, and CS Genetics is single-cell RNA-seq data. All of these have different chemistries, and then we wrote a bunch of evals. I'm going to talk about what evals are in just a bit, but then before then, and then we tested how these tasks performed on a variety of state-of-the-art models from Anthropic, OpenAI, XAI, and Gemini. So at a high level, these models are showing steady improvement in their ability to analyze single-cell genomics data. And then, the general statistical analysis that they're able to do with precision is something that is already impressive. But then our study also shows that there are certain aspects about single-cell data analysis that these models miss, which could be specific to kits, or which could be specific to way it handles a single-cell analysis task, which I'll talk about in a little bit. But then at a high level, we also saw that across, as these models evolved, they got better and better in their ability to be able to do single-cell analysis. For example, in terms of accuracy, we saw that there was a nice boost in Gemini's ability to analyze this data. We also saw a moderate improvement in Opus's data analysis skills, and GPT was comparable between 5.4 and 5.5. And in terms of latency, we saw that there was a nice jump in the latency versus accuracy trade-offs for GPT. Pretty much similar for Opus's between Opus 4.6 and 4.7, but then we also saw that the latency did go up by a lot for Gemini. But then this needs to be taken into consideration along with the accuracy that these models were able to bring in. And we also investigated if there are kit-specific artifacts in the bioinformatic capabilities, and we clearly see that the variants can still be broken down across these kits. For example, if you see Tapestry, which is right there at the far right of this graph, the model accuracy is pretty much low compared to the median performance, and this is primarily because this is an entirely different kit. This is a single-cell DNA kit, that has very specific needs in terms of the problems that we are trying to solve with this kit. And this also kind of shows that a lot of these models have been exposed to single-cell RNA, and then there's a lot of work that needs to be done with regards to analyzing DNA data. One thing that still I would like to double-click here is that we've been comparing this across different data sets with different complexities, and those are not really comparable across these models. So they could also be that some of these are more complex data, but then despite that, we still see kit-specific artifacts we were able to observe when we analyzed the trajectories from a lot of these models. Right, so one thing that I've been often talking about right here is that, okay, well, we designed evals. We designed, at least based on the previous slide, about 200 evals. But what are these evals, and how do you know an eval is actually good? So when it comes to designing these evals, there are three key design principles that we almost very sincerely stick to, and the first one being durability. So what makes an eval durable, right? Because when it comes to analyzing these data sets, the same inference can be reached through multiple means, and we need to make sure that the eval allows for variation in methodological choices, or that it shouldn't be that it works only with specific parameter choices. So it should have this room for all the right combination of parameters, and still admit these values as opposed to ones that do not make sense. The eval must also be anti-shortcut, or leak-proof. It really should not disclose the answer. Whatever we're trying to test, Essentially, let's say, for example, if you're trying to test an agent's ability to do cell typing, you shouldn't have a column called cell type annotation in your data set, right? That defeats the purpose of basically the model can just cheat. Or sometimes you can also see that you would be too procedural and you describe the entire method, allowing no room for improvement or like no room for reasoning for the model. So this is what we refer to as anti-shortcut, which is a second key design principle. And the third one is that it's verifiable. So independently, if I give it to a scientist, they should still be able to recover the ground truth and the tolerance values that we've set for these evals. So essentially, the numbers shouldn't be arbitrary or should not really depend on arbitrary choices for that particular eval. So using these three design principles as governing principles, so we've designed about 200 evals across all these models, across all these kit families, and then we tested these on the state-of-the-art models. So now, I've summarized a lot of these statistics before, but then I'd like to take a deep plunge into a couple of these evals that we thought were very interesting, and we'd like to present to you guys, and then, really show you where the model differed from the actual ground truth itself. So the first one is I would like to start with Tapestry, so which is a single-cell DNA kit, and I would like to expose the reasoning gap. I'd briefly describe what the eval does, and then from there, let's see what the ground truth looks like. So what we queried the model was we basically gave it a Tapestry data set, which again is a single cell DNA and a protein. It's got the protein immune panel. And then we basically asked it to identify the somatic DNMT3A mutation. And then look at the mutant fraction between NK cells and CD4 positive T cells. And all it had to do was compute this and then report this ratio, and then probably answer a biology question based on what this ratio actually means. Let's see what the agent actually did, right? This data set had 5 samples. So essentially, there was one sample where this particular mutation was a germline one, and there was another sample where this one was a somatic mutation. So the test is really if the agent can really look at the data and say, well, this is somatic and this is germline, and then compute this fraction. But then when we actually tested this on the on 16 episodes, or 16 different initializations of the same eval, we found that 11 of them failed because they were not they picked the sample that had the germline variant and not really the somatic mutation. And 4 of them failed because they got the ratio right, but then they were not able to resolve the biology. So essentially, in this case, it actually got the method right because it was able to identify the cell types correctly. It was able to look through the metadata and understand what fields were present in the data set. But where it failed was its inability to really distinguish between germline and somatic mutations with a lot of conviction. So now this is a very clear failure mode in terms of an agent's inability to really process discern between a germline and a somatic mutation. Another example that I have for you is where it performs the computation right, but then it kind of misses the context on why we have the agent perform a specific computation. This is from a PIP-seeker experiment. So this particular study that we were looking at had both single cell and single nuclei RNA-seq data. And for each of these data sets, we wanted to estimate the percentage of cells that are genuinely neuronal. Essentially, those are sensory neurons. And we wanted the agent to report this as a true fraction, a true neuron fraction between the two kits, right? So it's pretty clear. You'll have to do cell type annotation and then report what fraction of the cells are true neurons. But the lesson is that what the agent used was SNAP25, which is a textbook neuronal marker, but then it also did not account for ambient RNA contamination. What a domain biologist would have probably done is when they saw that there was a high fraction of neuronal cells, they would have gone back and then independently tried another marker gene. So in this case, when we tried nuclear markers with RBFOX3, we were able to see that it was only about 1.6% that were neuronal. And that is kind of very clearly demonstrated by the UMAP that I have right here. So now, when we tried this on 3 different random initializations, we saw that the agent always fell to this trap, and it overestimated the neuron fraction in this data set, because it was just restricting to one particular marker and did not really look through the other markers. So now I'd like to pass it off to my colleague who has a few other examples.
Thanks, Hari. Cool. So just to recap, what Hari just mentioned is that for single cell bench, we captured a wide range of assays that have different variability in terms of performance, specifically for single nuclear RNA and single cell DNA analysis. They typically often require, different pre-processing analysis step compared to the traditional single cell RNA-seq. And then for traditional, the widely used single-cell RNA-seq kit, we have Chromium and BD Rhapsodies that are heavily represented in the public, benchmarks, as well as open source tools such as Surat, ScanPy tutorials, and widely used in publications. So our hypothesis becomes, for this greater exposure of these analyses in training data, they actually may contribute to stronger agent performance on standard single-cell RNA-seq tasks. So the next 2 examples I'm going to share is essentially expose the agent weakness in terms of these overrepresentation. So the first example comes from the using the 10x Chromium dataset. So for the task prompt, I give it a human 10x Chromium single-cell RNA-seq from small cell lung cancer tumor across multiple lanes from Chromium. And then the question is, ask it to perform pre-processing or appropriate QC step and characterize barcode retention. So this is what the data look like across different lanes. So if you actually look at the data itself, we actually form a bimodal distribution. So one of the lane actually contain 40K cell-called barcodes, and on average, the other lanes actually have around 2K to 6K range. So this, essentially the 20K cell-called barcodes, they're actually ambient droplets. That's very specific to 10x Chromium kit. And so my hypothesis is whether the agent first is able to look at the data distribution and then make, reasonable judgment in terms of the threshold. So now let's look at where the agent fall. So all 3 agents actually detected a bimodal distribution, but it actually still applied a single global QC threshold. So in this case, it should actually look at the distribution of each lane and then decide, what is the most appropriate threshold, and instead of just do this one-cut-fits-all approach. And one of the run actually utilized the ScanPy tutorial default, which preserved 99% of these ambient droplets. And the vertical line you see here is essentially a ground truth we set, where it should filter out most of the ambient droplets. And then this is another look in terms of the data distribution. So for a scientist who look at the data the first time, to them it's like a big red flag, right? So this is very surprising that agent just used this tutorial approach to handle the data. Okay, so the second kit I choose is the BD Rhapsody. So this is slightly different from the 10x Chromium. Essentially, 10x Chromium, sometimes I think the data I pick is the whole transcriptome. For this case, it's the targeted panels. So it includes around 300 genes. So for this, I want to test if the agent has this panel awareness to perform analysis. So the task prompt I give is, given this single-cell RNA-seq from baseline triple negative breast cancer monocytes, and identify the top 20 transcriptional shifts between responders versus non-responders after treatment. And so the trap here is that because it's a panel-based data set, not all the genes in the panel are actually expressed in the samples. So in this case, there are a bunch of genes that just have very low noise level expression across all samples. And then what it did, so I want to test to see if the agent is able to pick up the panel-based panel signal, and then see if it's able to pick a good reasonable statistics to actually rank these top 20 genes. So if it just use the raw log2 fold change to compare, then obviously some of these very noise level genes will be inflated in terms of the fold change. And what the agent did is actually fall exactly into the trap where it didn't look any in terms of the expression across different genes. What they did is just rank all the genes based on the ScanPy log fold change function, and then just pick the top 20, which essentially are just noise and not like very expressed across all the samples. So essentially, it failed to capture the real monocyte signature. Okay, so those two examples actually show that because there are a lot of overrepresentation of these like 10x chromium or Surat single cell ScanPy based tutorials, which influence the choice of the, for example, QC. So now we actually look into the accuracy breakdown by task category. So in this case, for a single cell bench currently, we have, for example, we break evals into QC, dimensionality reduction, clustering, cell typing, and differential expression. So our hypothesis is that while agent typically actually would fall miserably in the more downstream analysis, such as cell typing or differential expression, because they require more like domain biological knowledge. But from our data, interestingly, you can see that the QC actually show lower accuracy. I think that's actually expected just because the nature of SC bench have included different modality in terms of assay types, right? So they all require their specific quirks in terms of the QC processing, which shows that agent doesn't have these like kit awareness to apply sensible QC for these specific kit. Cool. So with our current design principles of SC bench, so they are designed into these modular categories, as I just showed, so that we actually can be more, can essentially test these agents' behavior in a more controlled way. So for example, with the QC, clearly we can just ask it to perform QC and see how many cells are filtered out. And then to cell typing, then we just give it a clean data and then ask it to do cell typing to see if the cell types are sensible. But this also comes with its limitation because ultimately we really want to test agent's ability to kind of answer the big biological question, right? Given raw data, can you perform end-to-end analysis, pick sensible approaches, algorithms, parameters to derive the actual correct biological insights? So this actually motivates us to work on single cell bench V2, which is focusing on the long horizon evaluation tasks, where we give the agent the raw task and ask it a very high level biological question to see if it's able to recover true signal. So this is just a little bit flavor in terms of examples. So what the eval would look like is that you just give the agent a clean, for example, or messy cell by gene matrix, the raw data, and then you ask it, do cluster T cells enhance tumor killing versus singlets? So this is a very high level question, right? So we can actually break it down even more. So then to study the mechanism to understand this question, we can ask something like, so are APC conjugated T cells more exhausted than tumor conjugated T cells? And then break down even more to other different questions. We can actually test the analysis steps. So this type of questions come with its own challenge. So with the breaking down into modular category approach, we most of our evals actually use the numeric metric as grader to grade whether the agent did the right thing. So in the QC example, I just ask it to perform QC and see the number of cells that's retained, right? And then see if it makes sense, depends on tissue type or data. But then since now we frame our question in a more high-level, open-ended way, now how do we grade this becomes slightly more challenging. And what we found is that with the numeric grading method, this actually is also very challenging in terms of setting the ground truth. Because first, you gotta find a good differentiator that can actually have a strong signal to show you whether the agent did the right thing or wrong thing. So for example, we're looking for signals like fold change, right? If the agent did the wrong thing, then maybe the fold change is like inflated or vice versa. So with that, how do you set a tolerance window? Now we have agents help. You can actually try to ask it to sweep different parameters, combination of tools. But then if you pick the wrong differentiator, then maybe it's very method variant. So different methods might give you like different answer, which makes your eval not durable. So now with this grading system for the long horizon task, we're more thinking about to use more controlled vocabulary. So instead of grading it purely based on numeric numbers, can it actually answer like final scientific conclusions using these more controlled vocabulary. So this is just still work in progress. So we're still thinking about how to best set up graders for each of the questions we're trying to ask. So yeah, still work in progress, and stay tuned for the exciting results. Okay, that's it from us. And thank you for staying till the end, and we're happy to take any questions.
I guess it's a big set that to the protocols and check at the end whether the results published by the agent correlate with the results published in an article.
Yes, exactly. So that's actually what we've been doing. So for all the questions, all the evals we currently built, or to test the agent's performance, we trying to anchor it based on publications. And then that actually comes with its own challenge, because a lot of these like publicly available data can also be messy. Some of the paper results are not reproducible.
Yeah, we've all been there where a reviewer too wanted us to add a figure. So like, do you take that? Do you test on that or not? Again, it's a judgment call.
Yeah, so what we've been doing is actually trying to reproduce from our end, perform end-to-end analysis to see where's the gap between our analysis versus agents and authors' analysis, and find the ground truth.
This is great. And for the best case scenarios, what is the reproducibility rates? Do we have a...
Right. Yeah, that's a good question. So in terms of reproducibility, I think that's also kind of tied to the long horizon task, where the downstream ultimate science is pretty similar. It's up to like how strong the signal is, or like the intermediate step. You might get different QC like QC cell counts, right? But ultimately, it doesn't really affect your downstream results.
It again ultimately depends on that particular paper we are looking at, and so we do try to reproduce all the results. And if it doesn't convince any of us, we just that's questionable, and then we move on to the next one.
Yes.
Thanks. This is a great talk. Super awesome work. How do you guys think about assessing the difficulty of the task? Right. So, like, if you're trying to, for example, do cell type ID on PBMCs, which is way easier than doing subsets of T cells, right? So, how do you guys kind of parse that?
That's a brilliant question. Like, for example, brain is overrepresented a lot in the literature, so I think agents tend to get that right. But then on the other hand, if you're looking at pancreatic tissues or liver tissues, you have nice zonation patterns and things like that, and you kind of use that as guidelines too. So I think it really depends on what tissue you're looking at and what task you're trying to do at the end of the day. For example, I would say the DNA data that we analyzed, although it's a very simple assay, it's a hard problem for an agent, because it does not have a lot of things that it could have access to, and it should rather think logically before it tends to answer a question. So I think a lot of these times, it really depends on how well a particular task is overrepresented and how, if a model can simply do it, without a lot of help.
And on top of that, like for cell typing, right, itself is, like there's so many ways. Maybe they're marker-based, there's like reference, label transfer, can you pick the right reference? I think we just open a new avenue. In terms of self-typing, there's different level of difficulty, as you said. So this is definitely something that, like with long horizon tasks, is something we want to explore, is essentially, can agent utilize these, reference, pick the right reference for single cell to do these sort of more sophisticated cell typing tasks. Yeah.
I'm thinking that, isn't biology the only way you will assess it, in the sense that the paper, I'm saying something that I'm not sure it's true, but maybe using agents, you can actually, or an older paper, you can find results that they're more correct biology-wise, right? So is there another type of metrics you can have?
Oh, absolutely. So I think papers could be wrong, and they could be two papers that disagree on a single idea. So a lot of these times, I think there's definitely lots of gray areas when it comes to writing evals that are like super grounded in biology, and which is why we have a bunch of domain experts who we consult with when it comes to producing these evals. So yeah, I think that's a very fair point, and which is where I think you'll use your scientific integrity when it comes to answering this.
Create database resources that they've been created for years, but also be able to
Definitely,
manually.
yes, yeah. Yeah.
One more then. Will it be released to the community? Or is, I don't know, because it's private, right? Will you release a version or something, or guidelines?
The benchmark? Subset of it's available. We don't want the frontier labs to train on it.
Okay.
If you're using it for academic purposes, we can give it to you, and we'll just make you confirm you're not going to give it to them. That's kind of how that.
Non-profit?
Yeah, if you want it, we can give it to you. Cool. Thank you, guys. Thank you so much to Michaela and Valentine. And if you liked what, are you guys hiring? Well, you're not probably, because we're going to donate to you guys.
What do you need? Tell me.
And you're hiring?
We're hiring machine learning scientists.
So he's hiring machine learning scientists. We're hiring lots of engineers. This is not working, but we're hiring technical staff across many new domains. We use those benchmarks to engineer agents, and many of our customers are those kits that you saw. And we deploy those agents through those kits. So we're rapidly growing and looking for people to help us do benchmark engineering, but also a lot of the downstream engineering that comes from it, whether it's like building products, tools, RL, do that. So without further comments, I think we can close. We have some drinks over there, and let's just hang out and talk. Thank you.